In this lecture, we're going to focus on an important organelle that provides a very important function to the cell, and that is called the peroxisome. And the peroxisome, as you can see pictured here in this microscopic image on the right-hand side, are single membrane organelles. So they have a similar outer structure as the cell membrane, and they only have that single layer. The peroxisome does not contain ribosomes, nor does it contain DNA, but the peroxisome does have a number of different proteins as part of this organelle. So without DNA or ribosomes, where does this organelle get its proteins from? Well, these proteins come from the nucleus, and so the nucleus will initiate the process of protein synthesis, and it's a complex, multi-step array of assembling amino acids into new proteins, and those new proteins will be transported and incorporated into the peroxisome. And although there are many proteins, the one proteins of consideration here in this lecture are what we call oxidative enzymes. And so we see two such enzymes called catalase is one that I'll address here in this lecture. Urate oxidase. Once again, here we're looking for the suffix, the ASE, indicating that these are proteins that function as enzymes. And what enzymes do is that enzymes will speed up, catalyze chemical reactions by reducing what's called an activation barrier. By reducing the activation barrier, enzymes will accelerate a chemical reaction. One such chemical reaction that we will address here in the next couple of slides has to do with this molecule at the very bottom. So I know this text is pretty small, but this is an oxygen molecule. We have two of them bound together into molecular oxygen, which is the form of oxygen in the atmosphere that we need to breathe in to stay alive because we are aerobic organisms. However, this molecular oxygen molecule, O2, has this right here. This is not a typo. This is a negative sign that's supposed to be right here. But that negative sign here indicates that this molecular oxygen molecule has one extra electron bound to it. So that one bound electron changes molecular oxygen into a different form that we call a free radical. And a free radical is a molecule that does not have any unique purpose that supports the functionality of a cell or the organism, but is disruptive to the normal processes inside the cell. And we'll take a closer look at that here shortly. And so when we think about regular molecular oxygen, right, this is what we need to breathe in as humans to stay alive. Molecular oxygen functions inside a cell, specifically inside another organelle called the mitochondrion, by accepting hydrogen atoms. And that process will allow the mitochondrion, that one organelle that we'll get to, in making money, cellular cash, ATP. So in order to make ATP, oxygen needs to be pulled into the mitochondrion. It goes through a series of chemical reactions which binds hydrogen atoms, and in doing so, will make H2O. Now we call it H2O, not water. Water is a liquid, or water is a compound that exists in different states, but we just refer to this as H2O. So if we take 4 hydrogen atoms inside the mitochondrion and we bioreact that inside the mitochondrion with one oxygen molecule, doing the math, we'll end up with 2 harmless H2O molecules. Now, that occurs in the mitochondrion. This chemical process occurs 24 hours a day, 7 days a week. The magnitude of that reaction depends upon what we're doing. So, if we are engaged in physical activity acutely, as our metabolic rate goes up, the need to make ATP will go up. And as a result, this pathway will run much, much faster and to a much greater magnitude. And this is how we process molecular oxygen into water to make ATP in the mitochondrion in a perfect world. However, the cell is not necessarily a perfect world, neither is the mitochondrion. Oftentimes, these biochemical reactions do not run to full completion. So, on occasion, a single molecular oxygen molecule will pick up only one electron off of these four hydrogen atoms. When that occurs, O2 negative now takes on a unique name. That unique name is the superoxide anion, and this is now a free radical. This does not carry out the same function as molecular oxygen. In fact, O2 negative is damaging to the cell. It is highly reactive, which means it can form chemical bonds with a wide range of different molecules inside the cell and just disrupts the chemical structure of those other molecules. And if those chemical structures are disrupted, those molecules would not be able to carry out their natural function. So the peroxisome functions to absorb these free radicals and to neutralize these free radicals. And so the way it does that is that it will take the O2 negative and bioreact it with a hydrogen molecule, two hydrogen atoms bound to some other compound, and there's a wide range of other compounds that could be bound to these two hydrogen atoms that are bound together, but entering a reaction which is spontaneous inside the peroxisome, which means that you don't need an enzyme to do this. O2 negative gets what's called reduced to H2O2. This is called hydrogen peroxide. And hydrogen peroxide is much less reactive than superoxide, thereby neutralizing the potent damage caused by the O2 molecule, O2 negative molecule, rather. The next step inside the peroxisome is that the peroxisome will then bioreact hydrogen peroxide with another molecule a different molecule that contains molecular hydrogen to form two harmless H2O molecules. This pathway is not spontaneous, but is catalyzed by an enzyme just simply called catalase that originated from the intention to help keep the cell safe by neutralizing O2 negative. And this pathway is particularly important for liver cells and kidney cells and functions primarily to neutralize toxic compounds. A second function of the peroxisome is that it contributes to what's called beta oxidation. Beta oxidation is a term that refers to a complex pathway of metabolic steps to break up fatty acids. And so what we're looking at here on this slide is a saturated short chain fatty acid. So this is a fatty acid that we would call a 1, 2, 3, 4, 5, 6, 7, 8. This is an 8-carbon short chain fatty acid and is fully saturated as all the carbon atoms are occupied with hydrogen atoms. The right-hand side, the CHHH end, the CH3 end is referred to as the methyl group, and the carbons on the opposite side carry a particular designation. So you skip the first carbon on the carboxylic acid side of the fatty acid. This end is the carboxylic acid side. You skip the first carbon. The next carbon is called the alpha carbon. The carbon beside that is called the beta carbon. And when this entire complex is broken off of this short chain fatty acid, it is converted into a new molecule called acetyl coenzyme A. And this is a highly important molecule as it is used in a range of other bioreactions in various metabolic pathways. The third function of peroxisomes, depending upon the cell, is that peroxisomes will function to form what's called plasmalogens. And plasmalogens are a type of phospholipid that's found inside myelin, and I'll describe myelin here shortly. And so what we're seeing here is a typical neuron with a very long axon. And once again here, as introduced in an earlier lecture, we see this enlarged portion of a neuron that we call the soma, or the cell body, and that cell body houses the nucleus. The input of information to the neuron comes through these spiny extensions called the dendrite, and the cell body will eventually taper and narrow into a very long, what's called axon. So the axon that expands into these axon terminals are part of the actual same cell. It's just that this cell is very, very long. Now, in order to enhance the transmission of electrical energy from the cell body, or rather the tapered end of the cell body, to the terminal axons, that axon, which essentially functions as an electrical wire, must be or should be insulated if we want to transmit that signal efficiently and quickly. Myelin, which is made up of phospholipids, and lipids by nature are insulators. The myelin sheath will insulate the axon. This is not thermal insulation. This is electrical insulation, much like the electrical insulation around wires of various electrical appliances and computers and things of that nature. That's a similar idea here with the myelin sheath. And by confining the electrical signal to the axon, that electrical signal can travel much more efficiently and much faster to the terminal axon, particularly if we need to transmit an electrical signal to a muscle cell or to the brain or to some other organ in the body. So this ends this lecture portion of our unit on the peroxisome.